Hello, welcome to Chapter 3. This is our final chapter for our first exam. This is cellular form and function. We're going to look at cell types and cell membrane in this first lecture. Let's get started. So all organisms are composed of cells, from a single cell organism to a complex cells and tissue in the human. Cells are responsible for all structural and functional properties of a living organism. So if you think, what is a cell? A cell has everything it needs for an organism to live and to function as something. So the workings of the human body, the mechanisms of diseases, and also the rationale for therapy. So by understanding how cells work, you can understand how the body works. So let's look at cells. The study of cells is cytology. It all began when Robert Hooke, the same guy from chapter 1, coined the term cellare and described how empty cell walls of cork. So he saw the cork tree, he saw those cells of the cork, and he said, oh, those look like prison cells or different cells next to each other. So he coined the term cell and the study of them, cytology. Theodore Swan concluded about 2 centuries later that all animal tissues were made up of cells. And Louis Pasteur proved without any doubt that cells only arise from other cells. He refutes the idea of spontaneous generation, where living things arise from non-living matter. That might sound crazy, but they thought that bakers and people who made yogurt and people who made wine at this time. You probably wondered why monks and people of faith made wine and produced bread, because they thought it was a spiritual thing. They thought the conversion from the sugars in the grape juice into alcohol was magic. They didn't know there was something called yeast that made bread. So all of these things, Louis Pasteur had a couple of studies where he was able to say, milk doesn't spoil on its own. Some cells have to be in the milk for it to be spoiled. So he came up with the term pasteurization, and he was able to prove that if a cell is there, it starts to grow. It doesn't magically come from some type of religious or spiritual force. So no gods and no types of forest creatures created bread and wine. We're going to look at different cell shapes. So squamous, it's thin and flat. So the definition of squamous is it's wider than it is tall. Polygons look like a polygon. Stellates looks like a star. Another one that's red is cuboid. It is as wide as it is tall, so it's squarish, maybe even rounded on the corners. Columnar is taller than it is wide, so it looks like a column. Spheroid or ovoid are just round or oval. Discoid is disc. Fusiform is thick in the middle and tapered at the end. Fibrous looks like little strings. And some of these cell shapes appear in tissue sections, but not in their 3-dimensional shape. And that just means that if you're looking at squamous cells, and you look on the top, they just look like a layer, but here they're flat. When you look at cube, they look like they're wider than they are tall, and columnar, taller than they are wide. Those are polygons, star shapes of neurons, spheroid from white blood cells, discoid in red blood cells, fusiform in muscles, and fibrous connective tissue, which we'll look at when we do histology in chapter 5. And this is just another example of fibrous-like tissue. The human cell mostly is going to be between 10 and 15 microns. The egg cell is very large. A nerve cell, even though it's very long, it's so thin you can't see it with the naked eye. You have a lot of limitations on cell size. The cell growth increases in volume more than the surface area. All that means is the surface area is what a cell needs to get nutrients into the cell and to get waste out of the cell. So if you increase the surface area of the cell, Unfortunately, the volume of the cell is a proportion to the cube of its diameter, so the volume grows at an exponential rate when compared to its surface area. So the problem is as the volume of the cell increases, it's going to need more nutrients absorbed into it, more waste removal to get out of the cell, and because the surface area doesn't increase the same as the volume, cells have to have a limited size. They have many organelles. They're going to be divided into membranous and non-membranous classes. We're going to look at these in part 2 of these micro lectures. Non-membranous, they don't have a membrane. They come in contact with the cytosol. Membranous, they have a membrane that surrounds it and that separates them from the cytosol. So non-membranous, cytoskeletons, the microvilli, the centrioles, the flagella, the ribosomes, which we're going to talk about in part 2. We're definitely going to talk about the powerhouse with the mitochondria, the DNA in the nucleus, the rough and the smooth interplasmic reticulum, the factory of the Golgi apparatus, and the lysosomes and peroxisomes, how they clean up all damaged organelles and assist in cell suicide, or autolysis. The cell is going to have a plasma membrane. The plasma membrane is going to surround the cell and define the inside and outside of the cell. It's going to be made of proteins and lipids. Its composition and function can vary from one region of the cell to another. The inside of the cell is going to have cytoplasm. The cytoplasm can have organelles, the structure or the cytoskeleton, and also have intracellular fluid. It's going to have a nucleus and other types of membranes. Outside the cell is going to be the fluid that's going to be, or the structures and other stuff outside the cell. And the plasma membrane is made up of those lipids. You have hydrophilic heads that are water-liking, the lipids of the tails and cholesterol on the inside that fear water, and because they line up in 2 different layers, they're most likely going to be referred from this point forward as a phospholipid bilayer. 2 layers of phosphates and lipids. And this is the plasma membrane. Here you can see how the phospholipid bilayers separate the boundary of the cell outside and inside. You can then see the nucleus in here with the nuclear envelope. It has an intracellular facing towards the cytoplasm and an outward facing face. It can have anchoring proteins that attach it to the plasma membrane and other structures. It stops it from moving from side to side. It can have proteins that help the immune system detect it. Hey, I belong here. I'm a self cell. I am not foreign. It also has enzymes. These enzymes can be on the plasma membrane, so if something kind of passes through it, it can break down certain types of structures. It can also be a receptor. And if it binds to something, that binding is going to be a ligand, so some are going to be ligand binding receptors, which means a neurotransmitter or some type of chemical binds to a receptor, and that receptor opens, closing, or causes something on the inside to change. It can also be a carrier protein where something binds to it and then transfer them across the membrane, like in the blood-brain barrier with glucose. And then it can have channels. These can be a pore that just allow ions or small structures to pass through them. Here's your channel that can allow things go through. This is your glycoprotein with something on the outside showing, hey, I belong here. They can be transmembrane, where something can bind to the outside and cause a peripheral protein on the inside to do something on the inside. So the plasma membrane and all the proteins and all the other things that are attached in the plasma membrane are very important to how a cell works. And the phospholipids that we talked about before, 75% of the membrane lipids are phospholipids. The amphiphilic, they're arranged in that bilayer. That's why it's going to be a phospholipid bilayer. We talked about the water-loving and the water-fearing tails, and that's why they're able to line up in those 2 different layers. And because they kind of are fluid, they're allowed to drift laterally from place to place. And this allows things in the membrane to move around to where they want to be and create a fluid membrane. Cholesterol, when they're in the membrane, they kind of hold the phospholipid still and cause stiffening in the membrane. You can also have glycolipids or glycogen with lipids, and these are about 5%, and this helps to kind of maintain other types of things sticking out, like identifying self, or how the immune system can not attack some cells versus others. You can have some that are going to be transmembrane, and they go across the actual membrane. If they go across the membrane, they're going to pass through the membrane, they're transmembrane. The reason why transmembrane proteins are important is something can be on the outside, something can be on the inside. This is a very good way for something to communicate from the outside of the cell to the inside of the cell without actually passing the membrane. Also, transmembrane proteins can anchor it and actually hold it to different parts of the membrane. So instead of it floating from side to side, if it's anchored into its cytoskeleton, it doesn't move in that phospholipid bilayer. And we talked about peripheral proteins, just those are the ones sticking outside. So let's take another look. So what are some other types of membrane proteins? A chemical messenger. If something sits onto a cell, it's called the receptor. If that chemical messenger or something sits on top of its cell, and then that breaks it down, that's going to be an enzyme. It can be a channel that's either an ion channel that just allows things to pass through, or it can be gated, which means it opens and closes. This is going to be that carbohydrate on a lipid and protein. This is going to be a cellular identity. So it can be, this is A blood. I'm good. I don't need to bother with you. You can also have cellular adhesion molecules that attach it to other cell types to stop them from moving around. The receptors are how cells communicate, and they communicate via chemical signals. They can be hormones, neurotransmitters, but something binds to a receptor and communicates with inside the cell. That message can be a second messenger system. This is tricky. So when a messenger binds to the surface of the cell, it's going to trigger a change that's inside the cell that produces a second messenger in the cytoplasm. That chemical didn't go into the cell, but what happened, it changed that transmembrane protein, and the inside of it, with that peripheral protein, changed and caused it to be released, and that release is going to change something. Let's look at it. So a first messenger, like epinephrine, will bind to a surface receptor. This will trigger a change from within the cell to produce a second messenger in the cytoplasm. This causes the receptor to activate and release a G protein molecule. This is intracellular peripheral protein, and this guanosine triphosphate, an ATP-like compound, will then go and signal adenylate cyclase to convert to cyclic AMP, and cyclic AMP becomes the second messenger. In this case, cyclic AMP will activate a kinase. A kinase is a special type of enzyme in the cytosol. Kinases add phosphate groups to other cellular enzymes, and these enzymes can then go into the nucleus and cause gene transcription. These enzymes can also go to other types of parts of the cell, and what happens with second messengers is they trigger a wide variety of physical changes within the cell. But it's first a first messenger, and then that goes inside to a G protein. A G protein then goes to a cyclic AMP. Cyclic AMP then activates a kinase, and it's the kinases that are going to trigger changes within the cell. And here you can see first messenger, G protein, activates cyclic AMP. Cyclic AMP activates kinases that then either activate enzymes to various metabolic effects. Enzymes, when they're on the surface of the membrane, what they do is they just can carry out final stages of start protein digestion or break down certain types of neurotransmitters. They can also help produce second messenger, cyclic AMP, in certain cases. But pretty much, these are going to break down chemical messages and hormones when the job is done. It stops from excessive stimulation. Channel proteins are proteins with pores, and they allow water and other stuff and ions to pass through them. They're constantly open if they're a channel. Some of those can be gated, and that means they can open and close in response to stimuli. If it's a ligand channel, something chemical has to bind to it, and that opens the channel. Sometimes voltage within the cell changes from -70 to -50 to -20, and those different voltage opens and closes different channels. Some of them are mechanically open. So like if you stretch or you produce pressure, and those pressure allows walls of a laminated structure to touch, they can actually open channels, and it's that mechanics or that stretch or that pressure that opens and closes these channels. You can have a carrier or pump. So a carrier or pump is a transmembrane protein binds to glucose, electrolytes, and other solutes, and then transfer them across the membrane. A pump is going to use ATP. So every time you see a pump in the body, it's going to use ATP, and it's usually going to pump things from a low concentration to a high concentration, which is against their concentration gradient. And to move something uphill, you have to add energy, and in the body, it's ATP. We talked about the cellular identity markers. We're going to talk about this a lot more when we talk about the immune system, but it's pretty much your ID tag. And this enables our body to identify which cells belong to it and which ones are foreign invaders. And finally, the cellular adhesion molecules do exactly what they do. They adhere one cell to another cell or to the extracellular material. So these are all the different types of membrane molecules we have. Please go over them, because a lot of these are going to be used further on in this semester, and they start to be built on top of each other. So if you don't learn something now, sometimes it's really hard to catch up. So take your time memorizing these, even if you thought they were very easy. And good luck.